Media Advisory. Thursday, December 14th, 2023. Study suggests greater CD8 plus T-cell activity may increase HIV immunity. An effective HIV vaccine may need to prompt strong responses from immune cells called CD8 plus T-cells to protect people from acquiring HIV, according to a new study from researchers at the National Institute of Allergy and Infectious Diseases, NIAID, part of the National Institutes of Health, and colleagues. The study findings, appearing in Science, draw comparisons between the immune system activity of past HIV vaccine study participants and people with HIV who naturally keep the virus from replicating even in the absence of antiretroviral therapy, ART. The latter individuals are often called long-term non-progressors or elite controllers, LTNPs, ECs. When HIV enters the body, the virus begins to damage the immune system by inserting itself into CD4 plus T cells, which are white blood cells that help coordinate the immune response to pathogens. In most people, HIV continues to replicate and damage more and more CD4 plus T cells unless controlled by ART. Among LTNPs ECs, the immune system appears to promptly recognize CD4 plus cells with HIV and activate other immune cells called CD8 plus T cells. CD8 T cells destroy CD4 plus cells with HIV, enabling the suppression of HIV in a person's blood. The aim of an effective HIV vaccine is to provide durable protective immunity to HIV, or if initial defenses are bypassed, to help control HIV in the body long term, as happens with LTNPs ECs. Although several preventive HIV vaccine candidates have been designed to stimulate CD8 plus T-cell activity, they did not prevent HIV acquisition or control viral replication in clinical trials. Understanding and addressing this lack of effect is a scientific priority of HIV vaccine research. Scientists in the HIV-specific immunity section of NIAID's Laboratory of Immunoregulation and colleagues designed their study to better understand which CD8 plus T-cell functions were lacking in previous HIV vaccine recipients. They compared laboratory samples from previous HIV vaccine study participants with samples from LTNPs ECs. They found that both HIV vaccine recipients and LTNPs ECs generated large numbers of CD8 T-cells that recognized HIV. However, unlike the CD8 plus T-cells of LTNPCs, HIV vaccine recipients' CD8 plus T-cells failed to deliver the proteins necessary to destroy HIV-infected CD4 T-cells with HIV. Further tests suggested this dampened response was due to reduced sensitivity to HIV of vaccine recipients' T-cell receptors, the part of a CD8 plus T-cell that detects a CD4 plus T-cell with HIV. This reduced T-cell receptor sensitivity suggests the vaccine candidates from several prior studies did not sufficiently stimulate the maturation of CD8 plus T-cells to recognize, reach, and destroy all CD4 plus T-cells with HIV in a person's body. According to the authors, the study suggests that future HIV vaccine candidates may be more successful if they include additional doses or persist longer in the body to further stimulate the immune system. They also write that the potential of an HIV vaccine might be better judged by measuring how it affects CD8 plus T-cell function and sensitivity, in addition to just assessing the number of CD8 plus T-cells generated, which has been the usual practice. These findings build on decades of research by the HIV-specific immunity section of NIAID's Laboratory of Immunoregulation to better understand the immune response to HIV. The insights from this work may help guide future preventive and therapeutic HIV vaccine design and development, as well as HIV immunotherapy approaches. Editorial note: while the terms elite controller and long-term non-progressor are used in scientific settings, the HIV research community is working to identify person-first language as a possible alternative to these phrases. Miguelis et al. HIV vaccines induce CD8 plus T-cells with low antigen receptor sensitivity. Science DOI 10.1126 Science.ADG51 for 2023. Mark Connors, MD, Chief of the HIV-specific immunity section of NIAID's Laboratory of Immunoregulation, is available to discuss this research. To schedule interviews, please contact NIAID News and Science Writing Branch at 301-402-1663 or via email at niaidnews@niaid.nih.gov. NIAID conducts and supports research at National Institute of Health throughout the United States and worldwide to study the causes of infectious and immune-mediated diseases and to develop better means of preventing, diagnosing, and treating these illnesses. News releases, fact sheets, and other NIAID-related materials are available on the NIAID website. About the National Institutes of Health, National Institute of Health. National Institute of Health, the nation's medical research agency, includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nih.gov. National Institute of Health, turning discovery into health.